This should be a relatively short video, just a few slides left, but I didn't want to get them too long. Here we go, the metabolism for thiols and disulfides, so sulfur-containing compounds. They're really going to be analogs of alcohols, so very comparable to alcohols. So ethane thiol, you see here, is a sulfur with hydrogen. So we generally have thiols are going to have two bonds, and here we have just the same kind of idea as an alcohol. All right, so the difference is instead of just an OL ending, it has the THI, the thiol ending. So it's basically named the same as an alcohol, except for it's thi instead of just the all part. Just to So you know, these two compounds here and here, the skunk scent is caused by thiols. So a lot of sulfury kind of, that's why they, you can kind of imagine what hell would smell like. That generally they are referred to as sulfur containing or sulfury kind of scents. So not very great scents, generally speaking. Oxidation of thiols generally leads to disulfide bonds. So we're generally going to not have like a carbonyl type of groups. The thiols, the sulfurs, don't tend to make double bonds. So they just like to have single bonds. They prefer the single bond because it is a larger compound and it just has a hard time making those double bonds. So when we have a reducing agent, we break those those disulfide bonds, and we reduce them with reducing agents to create that thiol, that sulfur hydrogen bond. Then you might can smell kind of this when hair is permed, it's being reduced. So we break some of those disulfide bonds, and then new ones are formed, so that you can make curly hair. So if you are looking to have curly hair, you can get it permed. If you're looking to not have curly hair and break those up, you might want to try some reducing agent in your hair. Oh, one thing I did want to mention also here, disulfide bonds are important protein structural components. So a lot of times if you want to denature proteins, so we'll be doing this in our biochemistry lab, but we add reducing agent, and that's kind of the stinky stuff in some of those components that when we load protein gels, that's what stinks in there. It's that reducing agent that's going to break those disulfide bonds so we can make longer chain proteins that'll run out on the gel better and get, sodium dodecyl sulfate that's able to coat them when we're running those gels. Halogenation, the final kind of thing here we're going to go over quickly. Halogen-containing compounds, there are some. It's not it's not extremely common in biological compounds, but they're certainly there. And we do see this more. We've discussed this more in organic chemistry. These are usually have a prefix associated with them because they are lower priority functional groups. So they're generally given prefixes if it has a chlorine or bromine on them. You can see here, so 2-bromo 5-methyl hexane or trichloromethane. That's generally how they are named. One of the examples of biologically relevant halogen-containing compounds is, there's organic compounds are important for a lot of marine organisms. But few are significant in human biochemistry. One exception is the thyroxine, and it's an iodine-containing hormone secreted by the thyroid gland. So that's why we have iodinized salt. It's where iodine is added. So we don't have this problem generally in the United States because actually we probably eat too much salt. And so we get plenty of iodine in our diets. But other countries that don't have iodinized salt or they don't eat processed food, you can have a deficiency in iodine and have low thyroxine levels. So this causes swelling in the thyroid gland called the goiter, and you can see that on this lady here. That's why, again, that's added to table salt. So this is the thyroxine hormone, and these are the iodine functional groups that need to be added onto that for proper signaling and function of that. So I want you to look at this. There's a little kind of thought thing I want you to go through. They're stable. Halogen compounds are generally stabilized. So given this, hypothesize why the lack of halogenated molecule thyroxine would cause a goiter. So I want you to look at this slide right here. Given this image of thyroxine metabolism, can you hypothesize why a lack of iodine would cause a goiter in this tissue? So as you can see here, there are different components of the thyroid follicle. So you can see that follicle right here. We have cells on the outside of the follicle, and that's represented by this kind of blue thing here. We have a nucleus, and we have these things, and then a reservoir in the middle. And these cells here are going to bring in iodine from the blood and sodium from the blood, and they're going to pass it through into this reservoir here. The function of this cell is really to create this thyroglobulin secretion. So they synthesize these components. They're exocytosed into this reservoir. And in the reservoir, that's where there are enzymes that are going to manipulate these benzene rings and add on those iodinized components. Excuse me. And then once they're iodinized, then they're brought back into the cell, and then they're broken down into the thyroxine, and they're released into the blood system. So my hypothesis, and I would think maybe hopefully yours as well, is that what is happening, the reason if this doesn't have iodine, if we don't have iodine pumping in here, we don't have these iodine components put on, and then they're probably not endocytosed. So they're not probably being brought into the cell. They're not being received by the cell, because otherwise, if there's not some function or mechanism for keeping just this being inputted, then you're going to be wasting a lot of energy generating this and then just bringing it back into the cell. So your cell wants to make sure that it's been processed properly in this reservoir before it brings it in. So probably what's happening is we're getting this just kind of keeps pumping out, pumping out, pumping out. We're not getting any thyroxine, so your cells think you need to make more, hurry up, we don't have any, we need more. And then all of a sudden those cells begin to expand and blow up, and then you get this goiter formed. So that's basically, it's likely the cause of overworking the thyroid gland and causing that type of swelling. So that concludes our organic chemistry review. Hopefully you've enjoyed these videos because you're going to get a few more of these before the end of the semester. Okay, bye.